by kicking off this event and thanking everybody for being here tonight for day one of Drug Discovery Beyond the Bench, where we're looking at different industry careers from molecule to prescription, from bench to bedside. We hope that many of you in the audience will also be joining us for tomorrow's event as well. We will be covering different careers tomorrow than the careers that we're covering tonight. So for tonight, we have 5 amazing panelists who are going to be sticking around for the panel discussion and the speed networking afterwards. They are working in R&D, quality control, intellectual property, artificial intelligence, and clinical research management. Our 5 panelists for tonight are Deepa Balasubramanian, Jean Magram, Svetlana Pavlovich, Anshu Malhotra, and lastly, we have Krishna Oruganti. All right, so 5 panelists who you'll be meeting with tonight. I'm Thalyana Stathis from Memorial Sloan Kettering. I have a number of co-hosts here with me, Melanie Mahay and Matthew Bafudo, and Lori Hairview from INet New York City, and Abhi Day and Debanjana Chatterjee from STEMPeers. Okay, so now I have introduced everybody, all the panelists and all of the co-hosts. And we wanted to just say a few quick words about INet and STEMPeers before we launch into the panel discussion. Lori, do you want to just say a few quick words about INet before we get started?
Yeah, I'm going to share just a small PowerPoint. So welcome everybody. Thank you to the panelists who agreed to come and share their experience in their career path, and to all the attendees. So INet NYC stands for International Networking NYC. It was founded in 2014. Its aim is to support and offer professional development opportunities for international scientists in training in STEM in New York City area. And it's also its goal is also to help community building and facilitate connections between international scientists. So INET's board members are scientists from research institution in New York City. And we have more than 900 members that are foreign scientists in training. You can find more information on our website and become a member, or even apply for open positions. And if you need more information, you can follow us on different social medias or send us an email. Our upcoming event is in January 12th. It will be about science policy and diversity and inclusion. We would like to thank the INET board members and our advisory board, and of course our INET co-founders, Ariola and Mike, and also all of the organizations that have supported our events, like MSKCC, who is working with us today for this event, and also STEMPeers. So now I will pass the mic to Abhi, who will present STEMPeers.
Thank you so much, Taliana, and thank you, Lori. I'll give a brief introduction about our organization. Yes, welcome. First of all, everybody here, thank you for joining us. This is a collaborative event STEMPeers is organizing with MSK and INET, and this is the second edition of the event. So about STEMPeers, we are a global organization. I'll just quickly go through this timeline because it's been this year we're celebrating our 50th anniversary of existence, and it started as a social media platform for on similar objectives as INET, and we started having initial discussions about career development for STEM PhDs and postdocs in 2015, and with every passing year we developed on those discussions to create multiple avenues, not only for career mentorship but also for skill development. As you can see over here, from 2016 we started blogging from our organization through Club Sarai, and then we had by 2017 we started our first mentor-mentee program called Gurukul. as well as the STEMPeers annual conference that started subsequently, which blossomed into multiple initiatives for newsletter. Our European STEMPeer wing became existent in 2018. And we also started our Instagram channel, the STEM Times, which is very informative SciComm platform. You should find it out in Instagram. In 2019, we held our first collaborative drug discovery careers symposium. That was an in-person event last year with MSK, thanks to Taliana and INet. And in 2020, we are going further ahead with many other career development programs for undergrads, as well as for other as well as providing mental health support initiatives and diversity and outreach. So yes, in short, we are doing a lot of things, but we have several members of STEMPeers also attending today, as well as not only attending, they are also panelists over here whom you will see today. And with that, I would like to pass the microphone to Taliana for holding the poll.
All right, thank you, Abhi. Yes, so before we launch into the panel discussion, we want the panelists to know who all of you are in the audience. So I have launched a poll. Just take 2 seconds to answer these 2 questions. And then we'll share the results. All right, almost everybody has answered. Wait 2 more seconds. Okay, I'm sharing the results. So we have 55% of the audience is postdocs, 29% graduate students, and then 70% of the audience is based in New York City. Second most popular option is 17% are based somewhere else in the Northeast. All right, so that's who's in the audience today. We're so happy to see all of you. I'm now going to turn things over to Melanie and Matthew to start off our panel discussion. If any of you in the audience have questions, please put them in the chat. We will do our best to incorporate your questions into the panel discussion questions we are asking. If we don't have time, then please ask your questions during the speed networking. That's going to be your opportunity to ask all of those questions that we didn't cover in the panel discussion. And again, if anybody wants to turn on their camera, we'd be happy to see all of you in the audience. Otherwise, we hope that you'll turn on your camera for the speed networking in part 2. And now over to Melanie and Matthew.
All right, hi everyone. As Taliana said, I'm Matt Bafuto and I'm co-moderating this with Melanie. We're both members of INet New York City. And in the interest of time, I think we can jump right into the questions. So we want to start with each panelist just introducing themselves and asking, what did you research in your PhD and postdoc, your career path after you received your PhD or did your postdoc training, what your current job title is, and how did you make the decision to transition from your PhD postdoc into the industry you're in today. So we'll start with Deepa.
Hi, everybody. So I want to add a tiny bit more to this introduction. So in the sense that even before getting to my PhD, I've jumped careers and paths many times, or at least majors. I was a chemical engineer trained in India. I came to the US to go do a master's in bioengineering or biological engineering. And then slowly over time, from being interested in process and process development, became this person who was more interested in the genetics, the proteins, the biochemistry of stuff. And so I ended up, after almost 10 years from my undergrad, to start a PhD in structural biology at Purdue. And then I did a lot of work on cancer, epithelial cancers, with respect to structural biology, and learned a lot of mass spectrometry during my PhD. And from there, I postdoced at UCSD in biophysics and pretty much biochemistry. And then since then, I think I've stuck to whatever my core strengths are. 4 years after an academic postdoc, and after a lot of soul searching, I decided to go and test my waters, test the industry waters. I actually started my career in industry 5 years ago as a industry postdoc at Merck. Did that for 2 years. And then I'm currently a research scientist at Eli Lilly and Company in San Diego. And I transitioned from my industry postdoc to this position, and I've been here for 3 years. And I basically work on antibody drug discovery here in Lilly.
Thank you very much, Deepa. So next up would be Jean.
Hi, everybody. I'm Jean Magrum. I'm the Chief Scientific Officer at Celsius Therapeutics. And I started my career, I started my PhD, and finished my PhD at Columbia. I was studying in the Department of Genetics, and my PhD was on globin gene regulation in transgenic mice. And to tell you how old I am, we were the first group to actually show tissue-specific gene expression in mice. Now, that was a Nature paper back then. Now it's basically a supplemental figure at best. But then I went on from doing that to Mike Bishop's lab at UCSF, where I studied oncology. And I leveraged transgenic mice also for different aspects of studying oncogenes at the time. And then this question about career path, I then had the question, am I going to stay as an academic scientist and lead a lab in academia, or am I going to go to industry? And I didn't know what I wanted to do. And unlike you guys, I didn't have such a forum to hear more about what that was like. So I applied for both and went and visited both, and it went on lots and lots and lots of interviews. And in the end, I decided I really hate writing. So I didn't want to write grants for a living. I had done it to get my postdoc support, but I really love the idea of translational medicine and really thinking about leveraging scientific strategies to bring drugs to patients and do something that would be impactful for patients. And while that's of course true in academia, it's a little farther removed generally, not always, but generally. So I was super happy. I got a job at Roche as my first job, and I was working on right side by side with the guy who had discovered IL-12 at the time. And so we made a bunch of transgenics and knockouts, and I had a postdoc at that time, and he had a postdoc, and we together published a lot of papers on the basic biology of IL-12. So it was a great opportunity. As I mentioned, my current title is Chief Scientific Officer in a biotech, and I have moved from pharma at Roche to a biotech back to pharma, where I was 10 years at BI, where I 5 years was a group leader, and then for 5 years I headed the Department of Immunology. I then left there and went to Pfizer, where I ran CTI in New York, which is an academic industry partnership. And then I became Chief Scientific Officer of a company in Toronto, and then moved back to New York. And now I'm actually, the company I work for now is in Cambridge, Massachusetts. So I am based in New York. I live in New York, but my job is in Cambridge. And for a while, my job was in Toronto. So we can talk more about traveling. Why industry? I really wanted to make drugs for patients. I wanted to be impactful. I was excited to go do something that would be meaningful for patients. And that was my driver. And I really hated writing grants. So I didn't want to do that for a living.
Great, thank you very much, Jean. I think we can all relate to that sentiment. Next up is Svetlana.
Hi, everyone, and thank you for having me here today. So I am currently an associate lawyer at Aiken Gump. And prior to my current job, at some point I was a PhD student, like many of you. I never did a postdoc. So I did my graduate work at MSK, actually, in Robert Ben-Ezra's lab. And I used to go to many of, similarly to this, they were not Zoom events, but they were in-person events at Sloan and in Cornell, because this was a joint program between the two institutions. And so at some point I realized that I don't want to stay in academic science. And I was just interested in, everything else basically that was out there, which I could do, but that required sort of, my scientific background, that I could use my skills. So after going to many events over the years, it just somehow law really got me interested in. And yeah, it just resonated with me on many levels. I like that it's analytical. I like that I could use my scientific knowledge, that I would still be exposed to science. And so anyway, after my PhD, I then became a science advisor in a patent law firm, where I focused mostly on writing patent applications and helping inventors obtain patents. Now, I should say that during my PhD, I recognized that I will want to go down this path, and I decided that I will go to law school eventually. So then soon after starting to work in a patent law firm, I also started law school, which I did in the evening by working full-time during the day, sort of. So I went to Fordham Law in New York City. So I've been here for a long time, still in New York City. And after law school, I decided that I want to switch from patent prosecution, which is obtaining patents, to patent litigation, which is really enforcing and defending patents. And so now I work at a large international law firm doing patent litigation. So that's my path.
Great, thank you, Svetlana. Anshu, if you could tell us about your history first, please.
Sure. Hi. So I work as a clinical project manager. Actually, I recently switched fields from being a postdoctoral researcher studying children's brain tumors. So actually, to take a step back, I did my PhD in India, and that was a completely different field dealing with mitochondrial barcoding of organisms. And that was like almost 9 years ago that I completed that venture, and then sort of switched gears to come here to the US and then start looking into researching various aspects of cancer, which slowly led on to this pediatric tumor biology explorations. More specifically, I used to study metabolic and mitochondrial aberrations in brain tumors. And kind of towards the later half of my postdoctoral studies, I also got to explore drug development studies. And it was during that phase and the group that I was working on that sort of moment of realization happened, when I realized that I am very good at managing certain things. The keyword here being managing the research and projects and people. Not so much the bench aspects of science was what I was loving. And so working forth from there, it has been a journey where I like sort of sharpened my skills, where I realized like I was good at. And that sort of laid the foundations for my next step, where now I work as a clinical project manager for a healthcare informatics company. And so, yeah, I'll be happy to talk into more details of the different aspects as we go further.
Great, thank you, Anshu. And lastly is Krishna.
Hi everyone. So I'm Krishna, and I did my PhD in bioinformatics from University of Science back in 2010. So officially I was supposed to research bioinformatics, but I ended up researching a lot more than that. I was not really interested in bioinformatics per se. I did a lot of work on protein folding and machine learning, quite a lot of other things. And I would urge anybody who's still in PhD to use this time to kind of research a lot of things, because this is the last time you'll get when you can sit and just learn things. Okay, so I did that. Then for my postdoc, I went to University of Georgia, where I combined bioinformatics and biochemistry work. Okay, so we were studying protein kinases and their structural mechanisms. And at University of Georgia, we published a lot on protein kinase structural mechanisms from DNAs to Nature. But after that, I mean, I decided that I'm going to go back and look for faculty positions in India. And just as a stopgap measure, I joined this company, which was looking for a bioinformatics head in their company, GVK Bio. And I joined GVK Bio and I started working with pharma companies on pharmacogenetics work and several other computational biology work that happens in pharma companies. And somehow I really liked the pace of work. Okay, one thing I always felt in academia was it was too slow for me, but the company work was like, thick and fast. I mean, projects are flying off the shelves. So I really liked that kind of lifestyle. So at the end of it, I had an opportunity to join both as a faculty and also to transition completely into industry. And I decided to move to industry. And as part of my work at GVK Bio, we had also started a lot of artificial intelligence work using natural language processing to process all the number of papers that are coming out, and how to extract information from it, and how to use this for drug repurposing, or looking at the immunology landscape of all the papers, what is happening, how proteins affect each other, how proteins and small molecules interact. And so I specialized into more AI work towards the end of my first industry sprint. And at Genpact, I'm actually the vice president of data science research in pharmacovigilance and AI. So pharmacovigilance is drug safety reporting. So I would say that I'm at the junction of both the drug safety and regulatory practice. So we are helping pharma companies and regulatory authorities to process the drug safety reports that come in both during the clinical trials and also post-market analysis. So that's my contention.
Great. Thank you, Krishna.
Thank
Mel,
you.
do you want to ask the second question?
Yes, apparently we have a question for Jean from the chat. So someone is asking, what would be some example of starting position in pharma that would be recommended to someone who will be looking for an R&D job straight after a PhD graduation?
So I'll first start by saying my general recommendation is to do a postdoc if you can, because I think the more diverse your skill set, the more different kinds of things you know, you're more valuable to me when I hire you, because you have a broader perspective. So I like people who've done a postdoc. Now, I will admit, my daughter has a PhD in neuroscience and got a job in industry right out of her PhD, and she's an advocate for doing that. But in either case, whether you do a postdoc or not, generally your first role is at the bench, and that is it can be a number of different things. So we'll probably talk more about what drug discovery is and how you think about drug discovery. But generally, those kinds of roles are bench roles that give you the credibility to work on projects and learn the drug discovery process. And then as you get more and more experienced, you may manage other people, associates who are at the bench, and you may also start to lead projects and cross-functional teams that include members of those teams from different groups. So the actual jobs are generally not that different than what you do as a postdoc or grad student, and that you're designing, doing experiments, analyzing the data. You do much more teamwork in industry than you do in academia generally. You're much more involved, integrated into a team as opposed to sort of doing all those experiments and all that work yourself. But you're really working collaboratively with others to generate data that answers particular questions. And they tend to be more directed toward how do we decide if this project will lead to a potential therapeutic for a patient, and less about academic exploration of new biology. Not always the case. We can talk a bit more detail, but that's generally.
Yeah, thank you so much. Someone else is asking what would be recommended for entry-level starting position for recent pharma D grads. So it's a question for everyone.
I mean, I can start since this is a related question and everybody can chime in, but I think that is going to be extremely company and research dependent, right? So some companies need, bioinformatics or AI. We hire, like, for example, the company I work in now, I hire single cell genomics experts. I hire bioinformaticians, or we call them machine learning people, and other types of AI, systems, pharmacology, there's all kinds of roles in companies. And at the end of the day, it's your expertise. In pharma, we'll hire all of these roles. In biotechs, there'll be smaller numbers of these roles, but all of the work that you do will be applicable, and you just continue to learn new skills.
I'd like to add a little bit to this. I completely agree with Jane that you would, whatever your expertise is right now, you can use that to find a job if you look, but also rather than asking what position I should go into at this point, this is the best time to explore, talk to people who work in industry or whatever you're interested in, and figure out what you want to do, because whatever you choose, you're going to do this for a lifetime. Most of us are. You'll jump fields, you'll find other things to explore as you grow, but please take the time and learn about the options out there, just like from this panel or by talking to people one-on-one, and choose something that you like to do, rather than saying, hey, asking somebody, what should I look for? Look at a job ad and say, what skills I have, and will I enjoy doing this in some ways? You don't have to enjoy every little task in it, but I would look at this as a career rather than a, short-term job search and fill in, like, tick the boxes on every requirement.
Yeah, I think I would agree with both Deepa and Jean. Also, I mean, just to as a matter of interest, I can point out that even though we are doing AI work, okay, the major part of our work is in developing AI tools, but we do hire a lot of case processors who come from this B-Farm, D-Farm kind of a background. They come with a lot of experience, okay, in understanding the drug safety issues and everything. And I see that, pretty much on a daily basis, I consult with them on what should be your strategy going forward, what works, what does not work. So I would say that don't limit yourself only to pharma companies. You also have to keep in mind that this kind of domain knowledge is required not just in one industry, but pretty much across the industry. So even if you see an AI company, okay, just go back and see what exactly are they doing, and you can pitch yourself, okay, even if they are not thinking about it, you can pitch yourself that, this is what I would bring to you, okay, my domain expertise. And domain expertise is absolutely important to be successful in any enterprise.
Great. So I think we want to we're touching on this question, but to kind of go in deeper is, how did each of you learn about what's expected of a competitive candidate in each of your jobs after your PhD or postdoc? And how did you manage highlighting the skills that you already have with your willingness to learn new skills for a job in your future career? So maybe we'll start with Svetlana, because you had quite an interesting transition from a PhD to a JD. So
yeah, so I have to say I did not do much. Maybe I was just lucky. I think I did a good job just talking to people and during graduate school, and sort of confirming that this is, I think, the path that I would like to take. In terms of patent law, I don't think there are many internships available. One thing that I've heard people do is, for example, something you can maybe help on your resume is do an internship with a tech transfer office at your graduate school. I did not do that. So, it's probably helpful, but not necessary. Other people take the patent bar, which is something that you can take. It's a special bar for sort of patent attorneys, which is different than the regular bar exam. And what is interesting is that one can take it without having a law degree, as long as you have sufficient number of science classes. And again, that's something that is helpful that I did not do. So obviously, again, I'm just saying things that can help someone. I basically networked during my PhD. I then focused. One thing that I did do that was maybe helpful is I took the LSATs when I was applying for jobs. So I already knew going into interviews that I will be going to law school. I should say that going to law school is not mandatory. In order to have a career in patent law, one can become a science advisor, pass the patent bar, help inventors prosecute, obtain patents without having a law degree. But in my case, I already was applying to law school. I took the LSATs, and that was maybe a factor that was helpful to sort of just, show that I am serious about this career and very much so interested in it. I would say network and just apply to as many places as you can, reach out to people, and that's what I did. And somehow, I got a job. It wasn't through a connection. It wasn't through a contact. It was just, persistence.
Okay.
great. Yeah, I mean, as three organizations here that are sort of based on networking and heavy into networking, I think a theme we'll sort of consistently see throughout this is the importance of networking in whatever field that you may end up choosing. So after Svetlana, we're going to go through this question with all the speakers, and that'll be sort of the general format. And maybe at the end, we'll try and address, if we have time, some of the questions that are in the chat. So next, let's Anshu, if you don't mind.
Sure, yeah. I was actually going to pitch in kind of on the same lines as Deepa said. It's very important to discover your passion. And when you are in grad school or when you are a postdoc, that gives you a lot of different aspects where you can explore different things. It may not necessarily be targeted towards a particular job, but besides bench research, there's how do you manage if you have like students working with you? How do you explore your mentorship capabilities? If you are managing multiple collaborative projects, how does that kind of day-to-day scale? Are you able to juggle between different timelines? Different kinds of people with different expertise, those sort of things. There's a vast array of expertise that you could discover, or you could use that grad school or postdoc opportunity to figure out what you are good at and what you really enjoy doing. That I think matters a lot, and that is what played a very important role in my own career path as well, because I spent quite a bit of time in my postdoc, and it was only towards the last couple years that I realized what exactly it was that I wanted to do. And that realization came from what I was actually doing, because as I grew more senior in my postdoctoral ventures, there were more people who I got to manage. There were more and more projects being added to my work. So I was a little bit removed from actual bench work, but I was managing a lot of things. And at the same time, touching a little bit on the network part of, as you guys were mentioning, I have been a part of STEMPeers sort of since its very inception. And I had an opportunity, I would call it like an internship in a more informal sort of way, but I had the opportunity to manage their mentor-mentee program. I did it for about a year and a half, and I had no clue going in what I was getting into. But as I started, like with the help of the founders, as they gave me the larger outline of what was required from that position, and what were the people looking for as they came in as either mentors or mentees, that kind of gave me the idea that this is a part of my personality which I really enjoy. People management is how I had that realization that this is something that I could probably explore as a career. And so putting all those things together, things that I learned while being a postdoc and things that I learned during my networking sessions and during managing these mentor-mentee efforts, ultimately it all put together towards the end of my, like when my transition period actually started. And that sort of laid the foundation and gave me a clear, direct path to what I was looking for as my career, not just, my next job venture, but on a larger scale. So yeah, it's really important to figure out what you really like to do, and then take your grad school or postdoc opportunities to explore more things during that time period that you have.
Great. Yeah, thank you, Anshu. I think that's really important in order to say that although you're in a particular field, it's actually this multifaceted amalgamation of different skills, both hard and soft skills, and being aware of which skills you actually really enjoy utilizing and using, and then using that to sort of guide what you're passionate about into the future is great advice. So next, Krishna, if you don't mind.
Yeah. So since I had my PhD in bioinformatics, once I went to University of Georgia, I started collaborating with a lot of experimental groups on their computational biology requirements. And what I realized that most of the work that experimentalists are expecting from a computational biologist is very simplistic kind of analysis. Okay, they're not looking at very complicated stuff from computational biologists. Okay, because I don't think anybody really thinks that computational biology will solve any problems in biology. Okay, because experiments are what are important. So that realization made me realize that the deeper I go into bioinformatics, the farther I go away from the bench biology, and it's not going to be very useful for them, right? So that, and also the realization when I joined when joined GVK Bio, I had this realization that most companies again are not looking for very state-of-the-art kind of methods. They are looking at methods that are reproducible and which can be used day in and day out, which will provide mostly the results. Okay, so, and one part of that is real world evidence. Okay, so you can create all kinds of networks using computational biology or whatever, but it's not going to be very useful to pharma companies, because real world evidence matters the most. And I had absolutely no skills in real world evidence analysis or getting the insights from unstructured data and all those things. So I started building those skills. And one thing that really helped me was, after I left GVK Bio, I got involved in the startup kind of a environment where we were building these healthcare solutions in India, because India healthcare solutions don't exist. And thinking about the problems on how to develop these tools and technologies really helped me understand in depth what is required to do real world evidence analysis. And that made me much better prepared to take up the role that I have in Genpact, which is again analyzing real world medical reports and then extracting the drug safety information out of those medical reports. So it helped me understand the whole landscape much better, and it would be in a good place in getting this job.
Great, thank you very much, Krishna.
Thank you.
Deepa, I know you sort of touched on this, but is there anything that you would like to add specifically?
For me, the transition from academia to industry was more like, I think it was a choice I made pretty late into my academic postdoc. So when I started my academic postdoc, I was very sure I was going to be a professor. I was going to go out there, get grants. I put my heart and soul into my research. I worked very hard in getting a postdoc fellowship, which is not something that's, there are not many opportunities for internationals to get postdoc fellowships. So I worked really hard on it. I actually got a fellowship from the American Heart Association, and I thought that was the only path for me, right? But I also made some mistakes in that path, which are not very important right now, but I'm happy to discuss this aspect of it if somebody's specifically interested in talking to me after this meeting. But then life happened. I had a child, and I was not able to manage my own expectations, which is what we almost always fail on. I was not able to manage my own expectations of what I expected of me as a mother and being a postdoc and, searching for this career. So I started exploring other options. And that's when I transitioned to being an industry postdoc. The one thing that helped me, though, was this. During my graduate student years and my postdoc years, I took up a lot of extra stuff to explore things. My graduate school had a summer teaching thing for gifted children. I would volunteer and teach summer camps for 2 to 4 weeks while balancing research to explore and see if I liked, how did I like teaching? What did that, how did managing a course and coming up with all those things, how did managing children or younger students work out? There were a couple of opportunities similar to that when I was a postdoc. There was a company that had a small business grant that needed their technology evaluated, and they reached out. I don't know how it happened, but they came to my PI, and when my PI said, "Would one of you volunteer your time?" I said yes. And I just, I would say yes to every little opportunity where I could learn a little bit more, go and explore things, meet people. And so I expanded my experiences and my network. And so when I actually had to job search, like Svetlana said, I think I didn't apply to 100 jobs. I probably applied to 4. And I was very, very targeted. I would look, is this something I want to do? Is this a place I want to be in? Is this going to work out for me and my family? And I, pretty much chased what I wanted. And that's how I ended up in my current position too.
Thank you, Deepa. And lastly, Jean.
Yeah, I'll try to go quick, but I just want to say I appreciate all my colleagues on the panel's comment, but I'm going to be controversial because that's my nature as a scientist and disagree with a lot of what was said. So sorry for that, guys. And part of this comes from what I did when I was looking for jobs, but much more of my perspective comes from the fact that I've hired hundreds of people over the last few years. So, you know, first of all, I think you have to consider, I love this idea that you can, during graduate school, explore and then learn what you like to do. But there's two reality checks I would put around that. One is that that's hard. You don't know what the jobs in industry are. You don't know what job you'll get, and your job will change over time. So don't be too married to one thing that you love, because reality is, I will give you the example. I came out of my postdoc, I went into industry. My goal was to get off the bench. I didn't like, I liked science. I liked looking at data. I didn't like generating it myself. I wanted to lead a group. But you have to establish your credibility for yourself. So you need to start in a job that is on the bench so that you can show people you understand data, you can drive experiments, you really can be successful, and then you can move to the thing you really want to do. So I think if you know what you want, that's great, and you can go get it, that's great. I don't disagree with that. But I don't think you should be married to the view that you will identify the perfect thing in grad school, find it, and then you'll get that job. The other thing is my career went in and out of big companies, small companies, oncology, immunology, all kinds of different things, because I was in the right place at the right time in some cases, because I sought it out in some cases, because I got recruited in some cases. But reality is, you know, I wouldn't have predicted that career at the beginning at all, and I'm happy with the way it went. The second is when you start looking for jobs, I'm a believer in breadth versus depth. So that I think there are times where I need a technical expert in a certain area. We do single cell genomics in the company I'm at. I may need a single cell genomics expert, and that will be what I need. But usually when I'm hiring biologists or I'm hiring other people with other skill sets, I'm looking for diversity, because what we work on today might not be what we work on tomorrow. Company projects change over time. We evolve technologies and projects and all that. So I like people who think about different things and can connect dots in new and different ways. That's something I really look for. The third is that I can't overemphasize the networking piece. You know, I will tell you that for every job we advertise, we get hundreds of applicants. But if somebody sent me that CV and said, hey, you should take a look at this person, they're really good. Or somebody I met at a networking event, and I can tell you there's lots of them in New York. I go to them all the time. Well, not right now, obviously, but you know, you go to these events, you meet people, and you say, "Oh, I don't have a job for you now, but you know, 6 months later, I have the right job." I call that person up. So I can't overemphasize. I've gotten many of jobs I've gotten because I knew people and because I spent time cultivating relationships. And as a result, I knew it was a place I wanted to work because I knew some of the people and I understood what the culture was like. So that's really important. And the last thing, I'm just going to disagree with something Krishna said, that people are not looking for new technologies. That is not true in most of the companies I've been in. What you find is that in big companies especially, there's a wide range, and you're 100% right. If you're on the development end of the spectrum, putting drugs in the clinic, you need something very simple, very reproducible that can be done anywhere, anytime, and look the same. But on the very early research end of looking for targets and collaborating on academia, you may do something like Deepa was an industry postdoc. She probably did something that was publishable and something much more on the risky end, let's call it, not that. So I look for people with technology expertise, but generally what I'm looking for is people who are smart, they're adaptable, they were collaborative. I can't overemphasize the ability to collaborate with others and play well in the sandbox, because you work on teams. And all of that, I think, is extremely important in terms of how you think about it. So I do think it's great to know what you want early, but if you had asked me where would I land, I would have never said where I land now, 20 years ago or 30 years ago or however long it was. So just say.
Great, thank you very much, Jean.
Thank you
so
very
you
much.
sort of began to touch on this But my next question to the panelists is, what are your main roles and responsibilities in your current job, and how do those specifically relate to the drug development and discovery process? So I really want to get into the heart of the panel today, which is, how all of you in your various respective fields come together to produce a new drug, and what does that timeline look like? Or what does that process look like in your specific field? So we can start with Deepa.
Sure. So my specific role here in Lilly, the one thing I'm completely responsible for is epitope mapping, and looking at protein-protein interactions between our drug molecules and targets. I also do a lot of early discovery work where I look at the target ligand interaction. I have a structural biology background and also trained in hydrogen deuterium exchange mass spec. So my bread and butter is my science and my technology. So I was hired for my hydrogen deuterium exchange mass spec expertise pretty much for this particular job. So I work from any space in the very, very early. We have tens of molecules that have good biology. We'd like to look at their epitopes and understand how, mechanism of action, to this is our candidate, it's going to go into patents, and we need to know everything about its interaction on the antibody side and the antigen side, so that Lilly can know whether we are, I also analyze competitor molecules if they're out there already and published in patent literature or in publications. So we know where Lilly's molecule stands with respect to other molecules. And so that's pretty much how I also help in the drug discovery and development process. Because when you're making all these targets, you always want to know what they're targeting, where does the drug bind, what's its mechanism of action. So I go from anywhere in very, very early target space up to candidate selection, at least, sometimes after, depending on whether we get, somebody comes to fight us on the patents.
So,
and sometimes we do help development too, because sometimes molecules need some changes in development. So we go back and look at this kind of work to help them with formulations, etc.
Great. Thank you very much, Deepa. You touched on patents there, so I think we'll transition to Svetlana.
So, yeah, well, as I mentioned, prior to doing patent litigation, I did patent prosecution. And so, because I just want to say that there are really two sort of broad roles that patent attorneys are involved in drug development, sort of. And so the first part is during patent prosecution, we help, inventors, and it can be in academia or in industry, obtain patents. So that can be related to your drug. It can be related to processes, to manufacturing processes, and so various different aspects of drug discovery. Now, on the other side, in patent litigations, which is what I'm currently doing, we help either enforce the patents or defend them. But one role that we can have during the process is also, for example, we will get hired to assess for a company whether, let's say, they want to try and make, or they're planning on making a biosimilar, they would maybe ask us to assess the patent landscape, right? What will happen if they develop this drug, right? What would should they anticipate being sued by someone? What are the chances of, winning that lawsuit or not? So, we could assess the patents that relate to a specific drug, but what is also interesting is that you would look at not just the patents that cover the specific product, you would also look at the manufacturing patents, what kind of media is used. Maybe the media is patented, and maybe so you're doing that and you're infringing on someone else's patent. So I would say there are two broad sections, which is patent prosecution and litigation, and both deal with different aspects of drug development.
Great, thank you, Svetlana. Jean, maybe we'll go to you next.
Yeah, so I'm the Chief Scientific Officer in a biotech, so I'm responsible for all science from target identification all the way through putting things in the clinic. We are a single cell genomics platform company, so we are leveraging human samples in the context of clinical trials, plus minus drug, over time, responder, non-responder, all that. We can make comparisons using single cell genomic characterization of those samples. But ultimately, we're a drug discovery company, and while I can't explain drug discovery in 30 seconds, I'll just kind of make the point that I think there are jobs all along the way. So there are jobs in identifying targets. There are jobs in thinking about how you build a data package that's supportive of that target as a potential drug target. And that's called target validation. Then there's screens and characterization of molecules, what DEPA does. Once you have a molecule, after you've screened and you've picked it, how you develop the assays to screen through in vitro and in vivo and establish credibility for that molecule to do whatever your therapeutic hypothesis is. And then, of course, there's a whole lot of characterization that you never even thought about if you don't think about companies, where you have to do, as you said, formulation. Is it a pill? Is it an injectable? Is it a giant pill? Is it a tiny pill? All these things like that seem trivial to all of us are incredibly cumbersome and require a lot of expertise. So all along the way, you have to do tox studies in animals to make sure that your drug is safe. You have to demonstrate its efficacy. You have to predict what the dose will be in humans and determine how you're going to measure its effect in humans, not just on the clinical reads, but whether you've actually sufficiently drugged the target. How do you pick the right dose? There's all kinds of aspects of drug discovery. So my responsibility within the company is ensuring that we have a robust team that works together to generate new target ideas, test those hypotheses, develop mechanistic paths forward, and then biomarkers and strategies for going into the clinic. So all that's within sort of what my group does.
Great, thank you, Jean. Anshu, if you don't mind talking next.
Yeah, sure. So I actually, the company that I work for is not so much into drug development or discovery, but more into genetic testing. And so the products that we develop are more genetic testing products. And when COVID happened, we quickly repurposed, since we already had the infrastructure, we just quickly repurposed the lab. And now we are one of the major COVID testing laboratories as well. So in terms of my day-to-day or the major responsibilities in my role, I'm still quite new to this company, been just about 8 months in this role. But what I'm loving about it as a clinical project manager is that this role involves a little bit of everything. And so this is where I think what they liked about me and what I like about this role is that everything that I did during my grad school as well as my postdoctoral research, pretty much all different aspects are coming in handy right now in my everyday responsibilities. So there is a huge part of the whole process is next-gen sequencing, the wet bench processes that happen from start to finish. There are obviously bioinformatic pipelines. There is a fair bit of lab operations that I have to coordinate to make sure that in terms of equipment, in terms of everything else that the lab needs for their daily experimentation needs, that is all managed on a day-to-day basis. There is also some amount of dealings with genetic counselors, because once our final product is in the market and once we have started actually using it for patients, then how do we interact with our genetic counselors? So I am kind of in the center of all of these different aspects that are happening, and it is so it helps that I know a little bit of everything. I can talk to the research team in their terms. It's not hard for me to understand what they are trying to relate, say, for example, to the patient or to the genetic counselors. I can also form an interpretive bridge between the layperson who does not understand scientific terms so much, and then I can bring it back to the highly specialized teams who are working in the labs. And then what I did not do as part of my own research endeavors was I never dealt with bioinformatics pretty much like in the depth that they use now. But that's something that I learned on the go. It was a very steep learning curve. But then again, my biological knowledge helped. It's not completely foreign. It was a quick step into learning. So that's the whole part. Like there's so many different areas to explore and manage. And I just love that the team comes together sort of and converges at my role. And I am, relaying information effectively from one person to the other. And I think that's one of the more important things. I see there was a question also about my role and what are the skills that people look for. I think that management, of course, the name says it, but your ability as a project manager also includes a fair bit of people management and skill management. So that is the role that brings the most excitement out of this whole process that goes on every day in my life.
Great, thank you, Anshu. And then we'll finish with Krishna.
Yeah. So Genpact as a company employs about 80,000 people across different verticals. We work with financial insurance companies. We work with banks, with a lot of other companies. The specific product that we are building falls within the life sciences vertical at Genpact. And the product that we're building is on drug safety monitoring. So our work starts from the time a drug enters the clinical trials. So from that point onwards, whenever there is a side effect that you see in clinical trial, that needs to be reported to the company first, and then from there to FDA. Okay, so that process, during that process, a case processor, okay, who is a domain expert, usually reads that report and extracts all the relevant information about what was the patient age, what was the patient gender, what was the event, and was the event a causal link to the drug or not. All of those determinations, both biological and linguistic determination, case processors typically do this manually right now. Okay. What we are building is completely automating that process so that we are able to ingest the documents automatically, okay, and then read that, okay, using machine learning and AI techniques, and extract all the relevant information and be in a position to submit it to FDA or UDRA or any other regulatory agency. So this entire end-to-end process we are trying to automate. That's the product, and that's what we're building. And my day-to-day life is in managing a fairly large team. So we are about 8 data scientists who come from a different, both from computational science background, and also we have a lot of bioinformaticians who have transitioned to data science. We interface with another group of about 40 software engineers who are building the actual product, and that is embedded into a larger group of pharmacovigilance specialists and everybody else. So that's about 200 people who are involved in building this product end to end. And my specific task is to build the AI and ML routines that we need to use to automatically extract and process these reports. Okay, so my day goes into reading reports, okay, and then saying, what would a case processor see in a report? What are the subjective decisions that a case processor would take, okay, in extracting information? And how can I translate that into separate AI and ML algorithms? Do I need a classifier here? Should I add a regressor somewhere? Or should I add some kind of a NER system there, reasoning system? All of those decisions I need to take, and then build a system that will actually be live in different pharma companies. That's what.
Great, thank you, Krishna. Taliana, do we have time for more questions, or?
So I think I wanted to thank all of the panelists for sharing all of their insights so far. That was fantastic. And thank you to Melanie and Matthew for moderating. I think because it is 6:30, I'd like to transition to the speed networking part of the session.